Hello everyone. So today we are going to look upon important PYQ with solution from Unit 6 focusing on sensory photobiology. This unit encompasses a wide range of topics and questions comes from every corner of this topic, be it signaling part and signaling again, light and dark phase signaling, or it could be related to photoreceptors, specifically focusing on phytochromes, cryptochromes and phototropins. So, the kind of questions you can expect from here will be most of the times statement based only. So, they will be providing you with a combination of questions. You have to identify the correct combination out of that. So, let's explore the key questions and solutions that comes under this topic. So, the very first question is, young seedlings of Arabidopsis plants are exposed to the following light conditions: far red light followed by red light, far red light followed by red light, then dark phase, red light followed by far red light, dark phase followed by far red light and then red light. Statement E is about far red light being followed by dark phase and again red light. And the last statement is red light followed by dark phase and then far red light. So you have to identify the conditions that leads to photomorphogenesis. So they will be provided with you some combination statements, combination options. So the correct answer over here is option 4, statement A, D and E. So this question is basically related to the experimental condition with respect to seed germination, focusing on red light and far red light. So, statement we have, the correct statement is A, D and E. You will see that whether you will take A, D or E, everything is ending with red light, right? So, anything that ends with red light is considered to be the right combination when it comes to photomorphogenesis. Now, why it is so? Well, this was experimentally proven. As you can see, lettuce seed germination was carried out. Okay. When it was exposed to dark condition, there was no seed germination. When it was exposed to red light condition, seed germination was seen. Red to far red, that means first red light and after that exposing to far red, you will see that germination rate decreases. But after that, if you again put the red light, germination increases. But if you will put the far red light, again the germination rate decreases. So from this experiment, what was the conclusion is that red light and far red light has got the opposing effects when it comes to seed germination. So red light is the one that induces seed germination, whereas the far red light is the one that inhibits the seed germination. So if you are aware of this experiment, it is very easy to get a 4 marks out of this question. And not only that, this experiment, just a simple logic you need to remember. Red light is the one that promotes the seed germination. So this concept, if you keep in your mind, questions, anything related to this experiment can be easily cleared. Moving on to the second type of question. Directional growth of plants induced by light is called phototropism. Some statements are made on phototropism. Phototropism is a photomorphogenic response. FOT1 and FOT2 genes mediates phototropism. Cry1 and Cry2 genes although help to perceive blue light, but they are not involved in phototropism. Perception of blue light by phytochrome A photoreceptor initiates phototropism. So, it is also all about your phototropic event, that is movement towards the light. So you have to identify the correct combination of statement here. So accepting the statement D, rest all the statements are correct. So why the statement D is incorrect? See, perception of blue light by phytochrome A photoreceptor initiates phototropism. Let's check it out why the statement is incorrect. Always remember, phytochrome A is not a blue light receptor. Rather, it is related to red light and far red light. Phototropism, as I have already told you, it's a growth response of plant organs towards the light source. And this can be initiated by blue light perception. So for accepting this blue light, we have the defined photoreceptors known as cryptochromes and phototropins. So if you compare the three receptors, photoreceptors, cryptochromes, phototropins and phytochromes, what is the basic difference? Cryptochromes and phototropins are a class of blue light photoreceptors involved in phototropism, whereas phytochromes like phytochrome A are red and far red light photoreceptors. Although they are involved in various aspects of plant development, but they are not directly mediating phototropism in response to blue light. Moving on to the third question. Following are certain statements regarding phytochrome interacting factor, that is a family of proteins regulating photomorphogenic response in plants. PIF promotes scotomorphogenesis by serving as a transcriptional activator of dark-induced genes. PIF on interaction with PR gets phosphorylated, followed by degradation via proteasome complex. The degradation of PIF takes place in the presence of light. PIF-induced genes are not expressed in light. You have to identify the correct combination of statement here. So the correct combination of statement is statement A, statement C and statement D. So why the statement B is incorrect? Let's check it out. For this, you have to be very clear with the signaling, that is GA signaling in response to phytochrome. So if you look into the image, this is a dark or high GA that induces long hypocotyl. So in the dark condition, gibberellic acid is more, that induces the formation of long hypocotyl. So as you can see, phytochrome is present in the inactive form. So at that time, gibberellic acid comes and binds to their corresponding receptor known as GID1. That will be recruiting GRAS and DELA protein. We know that the ubiquitin, that is F-box protein, will come and bind to this GRAS and DELA. What it will do? It will add ubiquitin tags to the GRAS domain of the protein, thereby leading to proteasomal mediated degradation. So at this stage, what happens? The phytochrome interacting factor, that is the transcription factors, becomes free, which can go and bind to the G-box elements present in the gene via basic helix loop helix and carries out gene transcription. So that's the reason we say in the dark condition, high GA, long hypocotyl. Whereas in light condition, we have low GA leading to short hypocotyl. The reason is that phytochrome is present in the active stage, that is PFR stage. So when it comes inside the nucleus, it will target the transcription factor for proteasomal mediated degradation. Not only that, it is in the light condition, the DELA GRAS proteins are abundantly found. So they will be also targeting the transcription factor for degradation. So when transcription factor is itself degraded, how the gene transcription will be taking place? That's the reason in the low light condition, or not low light, low gibberellic acid condition, we have short hypocotyl. And this low gibberellic acid occurs in the presence of light. Next point. If you see in the light condition and dark condition. So what you can see is that we have phytochrome coming inside, which will be phosphorylating the PIF and they will be targeted for degradation. So the cell expansion genes will be inactive during the light phase. In the dark phase, what happens? We have gibberellin bound to the corresponding receptor. DELA will be recruited and it will be targeted for proteasomal degradation. At that stage, your phytochromes will move out of the nucleus. So what happens? Depending upon the requirement of the plant, it can either undergo cell expansion genes, that means the gene transcription of cell expansion genes, or it can block the synthesis of chlorophyll synthesizing genes. So that depends upon the requirement of the plant. So that occurs where? In the dark condition. So this is yet another diagram to explain the details regarding light and dark condition. Moving on to the last question. COP1 protein, an E3 ubiquitin ligase, regulates the turnover of proteins required for photomorphogenic development. Following are certain independent statements related to the function of COP1 protein. In the light, COP1 along with SPA1 adds ubiquitin tags to a subset of nuclear proteins. Proteins that are being ubiquitinated by COP1 and SPA1 can be targeted for degradation by 26S proteasome. In the dark phase, COP1 is slowly exported to the cytosol from the nucleus. The absence of COP1 in the nucleus permits the accumulation of transcriptional activators that are necessary for photomorphogenic development. Here also you need to identify the correct combination of statements. So the correct combination of statement is statement B and statement D. Now you need to understand what is the signaling pathway and how the signaling pathway is related to light and dark conditions. This is the image for that. So as you can see, we have the light condition here and we have the dark condition here. So during the dark condition, what happens? COP1 along with the SPA1 will be recruited inside, and these are the three transcription factors, HFR1, LAF1 and HY5. So in the dark conditions, they are not active, so they will be added with ubiquitin tags targeted for degradation. So at that time, phytochrome A also comes inside. During the light conditions, what happens? COP1 and SPA1 will be released outside. That means move out of the nucleus into the cytoplasm. Before leaving, it will add ubiquitin tags to phytochrome A. So they will be targeted for degradation. Because of which what happens? The transcription factors becomes free. So when the transcription factors are free, they will carry out the necessary gene expression required for photomorphogenic development. So as you have seen from this particular topic, majority of times statement based questions are coming. So you have to be very clear with every nook and corner of this topic. Then only you will be able to score well. And the fact is that every time you can expect questions from here, at least 1 or 2. And that too, it will be in the form of 4 marks. So never avoid this topic, and at the same time be clear with the every part of this topic. I hope so this video has helped you in understanding the questions they are asking in the exam, and now you can prepare yourself very well for this topic. Thank you so much.